So I have them here. So I have my samples, and then I have treatment and sequencing. Now we are going to set factor levels. So when we talk about factors, factors refer to conditions or groups. There may be slightly different definitions if you check other tutorials, but basically it's the same idea. So if you look at what we have here, the sample information, we have treatment. So for the treatment, if you check these samples, each of these samples will belong to either untreated or treated. And when we talk about the sequencing, each of these samples will either be single end or paired end. So these are conditions or groups. Okay, depend on, or how you want to define them. But then what you should take note is that because these samples here will belong to one of these groups, then the treatment becomes a factor, because we can look at differentially expressed genes between untreated and treated. So this becomes a factor. We also have sequencing. We can also look at differentially expressed genes in terms of the sequencing type. So we can compare single end and paired end. So sequencing also becomes a factor. So we are going to use that information here, and then DeSeq will run the analysis for us. So let's look at how to set the factor level. So in this tutorial, in this experiment, we have two factors. So we are going to do a multi-factor analysis.